Hello everyone, I am Madhav Subedi. In this video, I'll be talking about Hessian fly infestation rating in wheat. The main objective of this video is to learn about the identification and rating of Hessian fly infestation. In this slide, we can see various life stages of Hessian fly. Starting from the left, we have Hessian fly eggs that are laid on the leaves of a wheat plant. Then we have the larva, which hatches from the egg. Following it is the pupa stage of Hessian fly, and on the right is an adult Hessian fly which lays eggs and continues the cycle. The Hessian fly can cause severe damage to its susceptible cultivars. Its infestation can be identified from a few symptoms shown in the figure. In the panel A, we can see stunted symptom of Hessian fly infested plants on the right in comparison to the height of the uninfested plant on the left. The infested plants are prone to lodging as seen in the panel B. The Hessian fly susceptible lines will have thin width stand than the resistant lines. This can be seen in the panel C, where all of the brown plots are Hessian fly infested susceptible lines and the green plots are resistant lines. The Hessian fly infested plants also tend to be greener than the uninfested plants as shown in the panel D. These symptoms of Hessian fly can be used for rating of Hessian fly in the field and greenhouse setting. The other method of rating Hessian fly is by counting larvae and pupa in the infested plants, and this method will be demonstrated in this video. The materials required for the Hessian fly infestation rating are green cabbage bag, polythene bag, a tag, a pocket knife, a pen, and a paper. For Hessian fly rating, 30 to 40 plants per wheat lines are harvested from the field. The plants are plucked out and the attached soil is removed as much as possible. Plants from individual lines are kept in separate polythene bags along with a tag for the line. The polythene bags are then collected in cabbage bags and they are taken to the lab. If the size of the population is large, the Hessian fly rating can take a long time. Therefore, storage in the cold room at 4 degrees Celsius is recommended. Cabbage bags are taken out of the cold room for the Hessian fly rating. Individual polythene bag is then taken out of the cabbage bag. Any soil still left in the root is carefully removed without damaging the plants. 20 random plants are selected among the total plants harvested for that line, and the rest of the plants can be saved as a backup. The tag is also taken out of the bag, and it can be used for collecting the data on the infestation. Individual plant is then selected for the Hessian fly rating. We then split the plant in half from the base to the third node of the stalk. Now we look for the Hessian fly larva and pupa inside of the stalk. We also note for the Hessian fly larva and pupa outside of the stalk. The number of larvae and pupa in the plant is then noted down in the tag. Here, 1 represents the number 1 plant and 0 represents no larva or pupa found. The same process is continued for the rest of the 19 plants. Here is a close look at one of the pupa found among the plants for the same sample. There are some more pupa identified in the sample. The number of larvae and pupa is noted down for all of the 20 plants of the sample as shown in the tag. The data is then summarized in the front of the tag. Here 20 represents the number of plants, 6 is the number of infected plants, and 9 is the total number of larvae and pupa in combination. This data can then be used for association and mapping studies as shown in this figure. This concludes my description on the Hessian fly rating. Thank you for watching.